Have you ever thought about what it would be like to rewrite the code of life? I mean, not just to read the book of DNA, but to actually become its author. Well, that's exactly what we're talking about today. The incredible technology that turned what was once science fiction into a real-world scientific tool. Let's dive into how this all got started. It really all boils down to one kind of simple-sounding question. How do you copy and paste genetic information? You and I do it with text all day long, right? Copy, paste. But how do you do that with the molecules that make up a bacterium or a plant or, well, us? That's the big puzzle we're going to unpack today. You know, humans have been dabbling in biotechnology for literally thousands of years. Just think about it. Using yeast to make bread rise, or bacteria to turn grape juice into wine. That's what we'd call traditional biotech. We're using a whole organism to do a job for us. But what we're diving into today is completely different. This is the modern era, where we can go inside the cell, directly mess with the chemistry of DNA, and engineer brand new functions from scratch. This is the leap from being a farmer to being an architect of life itself. And like a lot of these huge scientific revolutions, this one didn't just pop out of nowhere in some big eureka moment. Nope. This is a tale of two brilliant scientists, each working on their own piece of a puzzle, whose paths were about to cross and, honestly, change the world forever. Okay, so our first key player is a guy named Herbert Boyer. Back in the late 1960s at UCSF, he was studying this really weird defense mechanism that bacteria have. He was totally fascinated by these special proteins that bacteria use to literally chop up the DNA of any viruses that try to invade them. These proteins were called restriction enzymes. But here's where Boyer had this absolutely incredible insight. He figured out that these enzymes weren't just shredding DNA randomly. Oh no. They were like super precise tools, cutting DNA only at very specific sequences. And the crucial part? The way they cut left these little single-stranded overhangs. He called them sticky ends, which is just a perfect name, because these ends naturally want to find and pair up with a matching partner. This discovery was the key. At the exact same time over at Stanford, we've got our second scientist, Stanley Cohen. And he was becoming an expert in something totally different, plasmids. Just think of plasmids as these little circular pieces of bonus DNA that just float around inside bacteria, totally separate from their main chromosome. Cohen had figured out how to get them out of bacteria, and even more important, how to get them back into other bacteria. So then comes this magic moment. In 1972, Boyer and Cohen finally meet. One of them has the perfect molecular scissors to cut DNA with incredible precision. The other has the perfect delivery vehicle to carry that DNA around. Can you see where this is going? Together, they had just invented the entire basic toolkit for genetic engineering. Okay, so they've got the tools, but how does this recipe actually work? I mean, how do you really use these molecular scissors and delivery trucks to copy and paste a gene? Let's break down the whole process. All right, first up, you've got your molecular scissors, those restriction enzymes Boyer was so obsessed with. Their entire job is to scan along a DNA strand, looking for one very, very specific sequence of genetic letters. And as soon as they find it, snip, they make a cut. So what is this special sequence they're searching for? It's something called a palindromic sequence, and it works kind of like a palindrome does in regular language. You know, a word like Malayalam. It's spelled the exact same way whether you read it from left to right or right to left. That's a perfect way to think about what's happening in the DNA. Just with a little biological twist. See, in DNA, the two strands have a direction. We call them 5' and 3'. A sequence is a palindrome if reading the top strand from its 5' end is the exact same as reading the bottom strand from its 5' end. So for example, GAATTC on top corresponds to CTTAAG on the bottom. If you read them both from the 5' end, you get GAATTC. This sequence right here is the target for one of the most famous restriction enzymes out there, EcoRI. Okay, so here's the really clever part. You take the gene you want to copy, let's say it's the gene for human insulin. Then you take your plasmid, your little delivery vehicle, and you use the exact same restriction enzyme on both of them. This is so important because it cuts them in the exact same way. It creates perfectly matching, complementary sticky ends. And because they're sticky, the insulin gene just naturally slots right into that open spot on the plasmid. Now, all you have to do is make that connection permanent. And for that, you need a totally different tool, an enzyme called DNA ligase. Look, if restriction enzymes are the scissors, then DNA ligase are the molecular glue. It just comes in and seals up the gaps, creating one single seamless new piece of hybrid DNA. And that is what we call recombinant DNA. And that plasmid, now carrying its new passenger, is our vector. It's our delivery truck. Its whole job now is to carry our desired gene into a host organism, usually something simple and fast-growing, like an E. coli bacterium. And this entire process, this whole idea, it all came together for the first time in 1972. Cohen and Boyer took a gene that gave bacteria resistance to an antibiotic. They used their brand new cut and paste method to pop it into a plasmid, and just like that, they created the world's very first artificial recombinant DNA molecule. A whole new era in biology had just started. But hang on a second. All of this stuff is happening on a molecular scale. It's completely invisible. How in the world could they know if their experiment had actually worked? How do you see the results? So yeah, let's tackle that final, critical question. You've done all the work, you've cut your DNA, you've pasted it into a plasmid, but how do you actually check your work and confirm you got what you wanted? The answer is this absolutely brilliant technique called gel electrophoresis. The idea behind it is actually pretty simple. DNA, as a molecule, has a negative electrical charge. So if you put it in an electric field, it's going to move toward the positive pole. You do this inside this slab of gel, which acts like a thick molecular jungle gym. The smaller a piece of DNA is, the faster and farther it can wiggle its way through the gel. It's basically a race where the smallest runners always win. Okay, so now all your DNA fragments are sorted by size, but they're still totally invisible. So to see them, you stain the whole gel with a special compound called ethidium bromide. This stuff is cool because it slides right in between the rungs of the DNA ladder and just hangs on. And then comes the grand finale, the big reveal. You put the gel under a UV light and bam, suddenly the invisible becomes visible. The ethidium bromide glows, and you see these beautiful, bright orange bands. Each one of those bands represents millions of DNA fragments that are all the exact same size, all neatly sorted out. This is your proof. This is how you see the code. And what's even cooler is that you can actually take a little scalpel, cut that glowing band right out of the gel, and then purify the DNA from it. That process is called elution, and it gives you a pure sample of the exact DNA fragment you need for the next step of your experiment. So, if we zoom all the way back out, this entire technological revolution we've been talking about can be boiled down to 3 main steps. First, you have to identify and isolate the piece of DNA you want. Second, you use this whole cut and paste method to transfer it into a host using a vector. And third, you gotta make sure that new DNA is stable in the host and gets copied every single time that cell divides. From making life-saving medicines like insulin inside bacteria, to engineering crops that can survive a drought, this amazing toolkit, born from that collaboration between Boyer and Cohen, has truly reshaped our world. And that leaves us with one really profound final question. Now that we have the power to write, and even rewrite, the code of life, what story should we choose to tell next?